This is the last lecture for the class, and I thought we could go through astrobiology. So when we say astrobiology, what do we mean by astrobiology? NASA defines it as the study of the origin, evolution, and distribution of life in the universe. So a nice narrow topic. Not. It used to be called exobiology some time ago. But they've changed that since, probably for PR reasons, to really astrobiology. And if you're interested further, you can read this astrobiology strategy that NASA published. They started publishing it in 2003, and it's the most recent version is 2015. So it was kind of interesting to read through, I gotta say. So if you want to pursue this further, be my guest and download that. It's interesting. But the thing about astrobiology is that there's a real focus on microorganisms. So why microorganisms? Well, as you probably understood over the course, that the world, our planet, Earth, is dominated by microorganisms since almost 4 billion years. So it is very, very possible that if you're going to find life on any other planet or moon or anything else, the most likely life you will detect is going to be microbial. And how is that? Well, aside from being very simple forms of life, in order to detect life, you also want to be able to detect global scale processes from afar using all different techniques, probes or radar or things, spectroscopy. And microbes, I mean, they really drive global processes on a planetary scale. And this gives us what we will call, or what the astrobiologists call, biosignatures. And there are many different types, and we'll go through that a bit. But I would say another reason is that microbes really, they're model organisms for really trying to understand how life began. So how did we go from chemistry to biology? And then what are the limits of life? And by having this model set of organisms, we can really start formulating and testing hypotheses on how life might be on other planets. If you remember this, I showed another variant of this slide. This is showing all the different types of cyanobacteria, which not necessarily 25% of all the world's cells in the ocean, but they are a lot. And it shows the different fluctuations of the different types of cyanobacteria in the different regions of the planet. So microbes really are a good way to try to figure out what might life look like and where might we see it. Hang on. There we go. But maybe it'd be interesting to, before we get into detecting life, maybe we should define what is exactly life, which is a really goofy question, to be honest. But people have attempted to answer that. And there's actually one series of lectures published by this famous physicist. You might recognize his name, Schrodinger, so of Schrodinger's cat. In 1943 or 44, he gave a series of lectures and published them, and it was called What is Life? Those lectures were given in Ireland, where he was a professor of physics at that time. And the way he defined life was that essentially it's a disequilibrium. And that means that things are not in equilibrium, things are out of balance. So it's this kind of equilibrium, disequilibrium, where you have an entity that is using energy to keep away entropy. So he uses very thermodynamic terms. And entropy, of course, means that everything over time in a closed system descends into disorder, right? So life could be defined as something that is using and taking in energy and taking in resources to keep away entropy, or what he called negative entropy. So the exact opposite of chaos and disorder, to keep processes in order. There's a few other things you can add into that. Another big definition of life that people toss around is that you have essentially molecules or entities, they can replicate and they can affect other molecules. There's this idea of a replicant. So anything that can replicate itself. And then, of course, it's using energy to do that, and it's affecting other molecules in order to replicate. So there's another bit of a definition of life that people have been tossing around. And of course, you can't really talk about what is life without mentioning evolution. Because over time, life also evolves. It changes as the environment changes. And then you get new variants coming up, older variants that are no longer fit for the environment going extinct. So you have these different definitions you can try to use to start and think about, okay, this is what life is, so how can we go about detecting it? So given that, what does life need? Well, we know it needs water, and there can be water in many different forms, but the water that life needs, it really needs to be fluid, it needs to be flowing water, because otherwise there's no molecular motion. You need to have the capacity for enzymes or molecules to be in a solution and interact. And for that, they need to be in a solvent of some kind. And water is the most likely one that you'll actually see life in. I think there's a few other hypotheses, but water, because of its special characteristics, being a polar molecule, it's the most likely that you'll see life. So we look for water first. So on this planet, there's been interest in, this is actually fairly recent, trying to figure out where did the water come from on Earth in the first place. Because like I said, you can have water in different forms. It can actually be locked up in the minerals. So just as like a crystalline part of the crystal structure of the minerals. And then as you have these sets of asteroids and you have a proto-Earth that's the accretion of all this different debris that forms our proto-Earth, you can have water that might be part of the geological minerals that's just from space dust, essentially. But in addition, you can have also bodies coming into the solar system that are over time colliding and then with our proto-Earth, and also bringing in other types of, also bringing in water. So depending on the types of asteroids, you can see they get different types of asteroids based on just their mineralogy and their rock formation. So you can have different sources of water coming into a planet depending on whether it came from the accretion disk over time as a solar system forms, or coming in from interstellar space and comets. But you need not just water, you also need all the elements that are necessary, of course, for life. And these are elements that have been formed from the overall fusion of hydrogen to start with, and then throughout, as a star ages, it's producing heavier and heavier elements throughout a star's evolution to give us the periodic table that we know now. But the biggest things, of course, is you need to have a planet that's going to be fairly rich in these particular non-metallic elements, our classic schnapps. Schnapps, I didn't even think about that till just now. That's funny. So, of course, we have carbon, hydrogen, nitrogen, oxygen, phosphorus, sulfur. But it's not just the non-metallics, the schnapps, that we need. We also want some metals as well, particularly our transition metals, because they can have a wide range of different redox states and valencies, and they can catalyze different types of reactions depending on just their valence state. So when you have potential catalysts interacting also with molecules that can form different types of compounds and bonds, I mean, that's what's so special about carbon, right? It can form 4 covalent bonds and form all different kinds of structures. But then in addition, you need energy. There needs to be an energy source from somewhere. So you can start off with energy coming in from the star, so in the form of light, but you can say a little bit differently. It's light, heat, but it's still something that's energy that's bombarding a planet. But with that, you also have to think about where might a planet be in order to find life, at least on the surface of a planet. So there is kind of this idea of a Goldilocks zone where you can have a potential habitable planet on the surface of the planet, depending on how far away it is from a star. And that, of course, is dependent on how big the star might be. So you can have different, like this, if this is our sun right here at a mass of 1, of course, if you have larger stars and different types of stars pumping out greater or lesser amounts of radiation, the distance, of course, is going to be, that's going to really have an influence on the distance that a planet can be in order to have surface life. If you're beyond that, or if you're closer, of course, you could have really, far too hot of a planet, and other factors, the amount of radiation bombarding an atmosphere. But if you're further away, you might not have enough energy. So then you start talking about a different type of energy that might be present and necessary for life, which would be geochemical. So then you're talking subsurface potential habitats for life. And even on this planet, we have a good example. But when I say geochemical, it's important to keep in mind that it's not that it's the heat necessarily that's coming up, that's the energy. It's the chemical reactions between things that are reduced and oxidized. That's the energy source. Just think of it as a battery. When you have compounds that are reduced and then you have other ones that are oxidized, there can be a potential for an electron transfer, just like in the negative and the positive ends of a battery. And it's being able to harness that transfer, those reactions, that transfer of electrons in order to get energy. The heat is still necessary because you still need to have fluid water, right? Because otherwise those kinds of reactions are going to be far more difficult to occur, as far as we know. I mean, maybe I should just add that too. This is as far as we know. There could be exceptions. If there's anything about biology, it's a science of exceptions. And on this planet, this is actually what's called I think it's called a cold smoker, but it's a hydrothermal seep that's in the mid-Atlantic ridge called the Lost City. And you have all this fluid coming up from the crust through a geothermal action, and then going up and forming these crystalline structures, these mineral structures deep down in the ocean. There is no light energy at all in this part of the ocean. So the only place that organisms can get energy is from chemical reactions down here. And you can have an environment that could be aerobic or it could be anaerobic, because you can still have oxygen and water. But if you don't, like the early Earth ocean probably was completely anaerobic because there wasn't any photosynthesis, you could still have enough oxidized compounds and enough combinations where you can still get an exchange of electrons. So one example might be if you're further out in the solar system, Europa, one of Jupiter's moons, has been a prime target for looking for microbes outside of Earth, because you have these what are called orbital resonances. And it's interesting because Europa is a completely ice-covered moon, right? But what happens is you get what is called tidal forces, where as Europa is orbiting in a slight ellipse, now actually it's a very slight ellipse, but it's still an ellipse if it's a very slight one, you have other moons like Io, which is closer in, and all the gravity between this whole system and Jupiter affects the moons such that you actually get like a stretching and squeezing of the entire moon, just like as the moon is going around the earth and the earth is going around the sun, you have the oceans, the tides increasing and decreasing, right? But you can have these kinds of forces where you're actually like stretching and squeezing the entire moon, and that can generate some level of heat that can actually keep water fluid further down below the outer ice crust. This is why on Europa, I should have put a picture in here, that was dumb, but you can see that there's all these cracks that they found on the ice crust because they believe that's what's happening. So you've got like an outer surface that's a little crunchy, but it's softer on the inside, and as you're squishing it back and forth, you wind up with these cracks, but you can wind up with a fluid system underneath. So there might be with some level of geothermal energy coming up. So it keeps the water warm and fluid, and there's probably some level of chemicals being barfed out into the water where the organisms might, any potential organisms could use it as an energy source. The last thing I think that really should be mentioned is that you need to have a stable environment for chemical reactions to occur. So you might have water and you might have chemicals, but if you have the whole planet being just blasted with the radiation, you're not going to have compounds that it's not going to be a stable environment for reactions to occur and then stay as the compounds, as the products. So Earth, of course, has a very strong magnetosphere, which helps shield us from all the solar radiation that would otherwise just blast away all the atmosphere. This is what they suspect happened to Mars. If you look at the magnetosphere on Mars, it's very weak. It died off a long time ago, and they're still working out why, but you really have a very thin atmosphere on Mars. I'll go into that in a bit. But that also leads to a very high radiation environment. So life on the surface is implausible, now at least. But we can also go back to this, what is life? And we can think about the big things, you know, these big picture things, but we want to think also about the second definition here about molecules that can replicate and affect other molecules. And what's interesting is to think about the jump from chemistry to biology is really rather substantial. And it's fun to have this in your head. So let's go from elements. So you start off with our schnapps and our metals. I forgot to include them here. You have to go from individual elements to some kind of organic molecule to form a particular compound or a structure that might be able to form longer chains of it. But to go from elements to organic molecules, well, there are potential sources ways in which this can happen. So, for example, in interstellar space, you can get these molecular clouds, these dark molecular clouds, and this is in the constellation Taurus, and they can detect that carbon out in space, you can actually get reactions amongst carbon molecules to form these aromatic rings with nitrogen attached to it, just through large conglomerations of dust that are also being hit with energy, with radiation. So in some ways, the radiation can help and form these compounds. So it's a little bit of a paradox there. So we can get organics from outer space onto a planet, or you can have all the elements on a planet and you can have them combine through different types of forces to make more complicated organics. So what I mean is not organics, elements. You can have all these different simple compounds that are not organic. So, methane, but you have ammonium, hydrogen, and water. But with the right conditions, you could actually get them to form more complicated organic molecules. This experiment might look familiar to some, the Miller experiment. So what was done, this was back in '53, is they just had a glass bulb and they had electrodes were there to put a current through it. And they fluxed in water and methane, ammonia, and hydrogen. and then shot in a little extra water here and heated it up and then electrocuted, essentially. So put in an electric current, like you would get from static discharges and lightning. And after doing this for a long period of time, I forget, it was like I forget how long they ran it. It was a couple months. So you get this accumulation of compounds where within that, there was a lot of different organic compounds. So these rings and different structures, and they found 5 amino acids. Then they found 5 amino acids that are commonly used in proteins now in life. What is more fascinating is they found 5 amino acids in 1953. Since then, the technology for detecting lower concentrations of amino acids has improved. They actually, when the guy retired, when Miller retired, they found the vials from this experiment, or a slightly different one, but almost at the same time, in his office, in his lab, and then they reanalyzed it. And they found 25 amino acids in 2008. And they said that you could probably find even more with even better technologies and more sensitive techniques. So you can get this buildup of organic molecules through rather fundamental and simple processes just on this planet, the building blocks of life. Not life, but the building blocks. But that's the thing, they're building blocks. They're what you would call a monomer, right? Just a one piece. So we have to think next, how do we go from monomers to polymers, multiple pieces glued together? Well, probably the most important reaction is what's called a condensation reaction between monomers, where you have one monomer here, another one there, and you get an input of energy so that they join up and form a polymer, and then you get a byproduct. For example, here's water. This is something to think about because this takes energy to do. So it's not something that just happens spontaneously by itself. You need to have the right concentrations and the right conditions. And even worse, if you have a lot of water, hydrolysis occurs spontaneously. So once you get a polymer together, then you have to worry about it breaking apart automatically and releasing energy. So how do we do that if we want to start life? One hypothesis that is also quite recent is it's through the mineralogy of the planet. For example, I won't go into detail, but a process called serpentinization, which winds up going from a stone called olivine, which is a rock that's in the deep mantle of the crust. Over time, it reacts and you get another stone called serpentine. You can actually buy this stuff and it can be used as stone that you put on the outside of a wood stove, a bras camin, if you want to keep it warm, like talistem. But the idea is that this process geologically actually takes in water and sucks it in so that you don't have too much water present. So if you have those big spires that you see, the mineral ones I showed you in the ocean, and then you have these processes ongoing, you can reduce the amount of water that's present to avoid hydrolysis. You can have a more concentrated set, a higher concentration of monomers, and they can get closer and closer together. Combine that with metals that might be able to react and catalyze reactions, and you might get circumstances that are thermodynamically favorable for condensation reactions and polymers to form. So over time, it might be that you get these chains of organic molecules building up, and then eventually potentially proteins, just through the chemistry of the rocks. This leads to potentially interesting polymers. For example, one of the hypotheses is that if you're able to get a lot of nucleotides, and they form RNA. RNA is actually self-catalyzing. It's a string of ribonucleic acids that will fold up and form these 3D structures that can actually have a certain level of catalytic activity. So they can bind onto things, they might be able to break apart things, and it's actually called a ribozyme. This becomes really interesting, and in fact, the hypothesis is that Earth started off as an RNA world. This is might be familiar to a few of you. You might have seen this in like popular science. But the other thing you have to keep in mind, it's not just because it is self-catalyzing and potentially self-replicating, it's holding information. You have different sets of nucleotides that can form different sequences and form different structures. So now you have a molecule that can self-catalyze and catalyze other reactions, potentially self-replicate. They've actually done this in labs, short pieces, but they've been able to show replication, and it holds information. It gets even more interesting when we start looking at what happens when proteins and RNA combine, because maybe the complexity increases. So we go from polymers, but then we have to contain them in something. So one idea is that, of course, lipids are also a type of polymer. And when you have polymers that are forming in these cavities, depending on the conditions, the lipids can form different types of structures. So everyone, you heard the term a micelle, right? So a lipid, of course, is a little polar head with a fatty acid or a fat chain. So if it's like an oil droplet, the fat chain will go into the oil droplet and then form an emulsion because the polar part is on the outside. And then that's how you can break apart oil droplets in water. But a micelle means that it's still oily on the inside. You can have vesicles that form where it's aqueous on the inside. And then you got this hydrophobic region that is surrounded by an aqueous region. So these kinds of things also happen just because of the chemistry of the environment. The pH can affect how these guys form micelles and vesicles, the mineralogy as well. So it could be that over time, you start getting these vesicles that maybe envelop these self-catalyzing molecules. And last but not least, one of the most important things is, as we said, life is about using energy to keep away entropy. So where might the energy be coming from? Well, we said it could come from light, can come from geothermal, but the organism still has to harness that. One hypothesis is that, and this is something maybe I should back up a little bit. This is kind of a universal feature in cells, is that we have these proton gradients across the cell. A proton gradient, all you have is a higher pH on one side and a lower pH on another side. When we respire, this is what our mitochondria is doing, is it's generating, it's using the electrons and generating this proton gradient, which the cell then uses to make ATP, what's called a chemiosmotic potential. What becomes fascinating is if we go back in time, geologically, these seeps, what happens is you have water coming up into them from the Earth's crust through geothermal reactions. That water is very alkaline, high pH, low amount of hydrogen ions. But the ocean water is a lower pH. So you get a natural gradient of protons, and you can have them in these cavities. So you wind up with a potential energy source that's, again, ready-made from the geochemistry of the planet. So this is one idea about how you might have evolved here, for example, in one of these small cavities over on the right. You have organics that are inside the cavity where it's a higher pH, pH of 9, and outside a lower pH where there's more protons, and the cavities act as a place where these things can react, where the carbons can start forming polymers. And as we go up in time, it might be that we evolve, okay, a particular structure that can utilize this potential where the hydrogens come in and do something. So because you want to get to an equilibrium, you have a potential energy source that maybe a molecule learns how to harness and creates and generates energy, or at least conserves energy out of that. So these notions of proton gradients that form naturally, that could be a way that organisms harness energy. So when we think about looking for life and we know what life can be like and what it might be doing, we start looking for biosignatures. So the idea of a biosignature is that it's an object or a substance or a pattern whose origin is specifically requires a biological agent. And we can kind of divide them up into three categories, I would say. So you have the compounds to look for, the organics. There might be inorganic compounds too that are only produced by life. This one has been up in the news recently on Venus, phosphine. So it's a phosphorus with 3 hydrogens attached to it. You can also look at ratios of isotopes of different elements, because the way biological organisms work is sometimes they prefer specific isotopes more than another isotope of an element. So they can change the ratio of the isotopes. But then you can look at minerals. So for example, these banded iron structures is when you started to see oxygenation on this planet, because as cyanobacteria started producing more oxygen, everything started to rust out because you had more oxygen. Iron was more reduced before then. There are also in the rocks, you might be able to find microscopic structures that look like bacteria or cells. We'll get into that in a minute. Or even larger structures. For example, this is a stromatolite, a fossilized stromatolite, which are cyanobacteria that form these large structures, like what you see in Australia and Shark Bay and some down in the Caribbean. Some of the earliest life on Earth that started to form oxygen. But there are also other signatures that you can look at from an even greater scale. So you can look at pigments, like the reflection off of a planet might give a certain spectra, chlorophyll would. Or you can have gases in the atmosphere and changes, like periodic changes in the gas concentrations in the atmosphere. And these biosignatures you can detect by remote detection. And this is how the phosphine, and phosphine, by the way, is thought to be only produced when you have decomposition processes of organic material that has phosphorus in it. So there was a big article where they thought they found traces of phosphine in the atmosphere of Venus. And they said that is a signature of life, or not. There's been I'll get into that in a minute. So you can have, and they detected this by looking at the spectra reflecting off of the planet. You can also have direct detection. So if you send a probe out that has detectors on it and can actually sample directly, this idea of Enceladus, a moon outside Saturn, it has these enormous geysers coming off of it. I'll show you that in a minute. But you can send probes through these kinds of things to see if there's biosignatures in the material being thrown off the moon. But the thing is, biosignatures are incredibly tricky, and you need to have certain criteria if you're going to call something a biosignature, a compound or whatever. It needs to be reliable, so low levels of false positives or negatives. It needs to be detectable, because maybe you have two processes that are abiotic and biotic producing the same signature, but one of them is producing it at a much higher rate. That means you can't detect the life. Maybe the abiotic process is producing so much of this biosignature that you'll never detect the biotic one, even though it's there. And they also have to be survivable. So it can't be super short-lived. So for example, a fossil, you can have fossils that are being preserved well enough where you can still see the structure, but maybe there are processes on the planet where those fossils are completely destroyed through whatever. It could be volcanic activity or tectonic activity. It could be a variety of different erosion through wind or sand. Like Mars in particular is very sand blasted. So you get a lot of erosion just through the sand whipping through and then striking minerals, at least on the surface. And then also in the atmosphere, if you have different compounds in the atmosphere, it might be that there's so much radiation that the compounds break down. So life is underneath the surface producing things, but as soon as they're up in the atmosphere, they're gone. So this idea about you have looking, like for example, oxygen is a good example. If you want to say, all right, this planet has oxygen, therefore it has life. No, there might be different scenarios in which oxygen is present, but there's still no life. So you have to be able to weed out based on other chemicals that are present or absent, for example, whether or not a particular signature of oxygen is a candidate for a planet that might have life. And this is a good example there's also the final element, I think it's the human error. So this was big news a few months back. They published in September last year, and they found phosphine on Venus. That is a signature that there might be life in the clouds. We know Venus itself has an atmospheric pressure of God knows how many hundreds of atmospheres and is 400 degrees centigrade at the surface because of this massive greenhouse effect. But Carl Sagan, a very famous astronomer, hypothesized that maybe in the clouds there could be all sorts of life that exists. And that's a good hypothesis. But what they found after this was published about the phosphine was that it's not phosphine, it's sulfur dioxide that they detected. They didn't analyze the data correctly. And there's now a lot of papers being published showing how it's not phosphine. So biosignature, sure, but you really have to be very, very careful about how you detect it and what it really means. So with that, how do we know if our biosignature is really, legitimate? Well, we go back to Earth again. And the thing about Earth is we do have a lot of habitats that are very extreme. And we say extreme from our perspective, of course, but you could say that given the average temperature and the average living conditions on the planet, these are very extreme environments. These are at the very edge. So really high temperatures or really low temperatures, really low amounts of water, pH, all sorts of different types of environments that we would consider quite extreme and might be analogs for moons that have certain geologies to them. But the thing is, we can access that. They are experimentally accessible systems. And there are a lot of different sites on the planet that are being used as analogs for finding extraterrestrial life because it's amazing that things even live there. But by investigating them, these really extreme environments, so for example, you have high temperature and submarine hydrothermal vents, soda lakes, high pressure, all these different things where extremophiles live, really low pH. So a lot of astrobiology is trying to find where's the limit of life. What is the very edge at which things can live? And if life is possible in these really extreme places, what does it look like? And in addition, with these kinds of habitats, you can test whether or not you have abiotic signals that look like life, but are not. And that is really important. A really neat example is actually here. This is called the Danakil Depression in Ethiopia. It is 120 meters below sea level, and it was nicknamed the Land of Death for a damn good reason, because it is what you would consider maybe polyextreme. So it's a massive volcanic rift. It's part of the Great Rift Valley. Excuse me. Much of the environment is hypersaline, so the water actually can range from 37 to 70% salt concentration. So super saturated salts. And the pH can range, it ranges from negative 1.6 to 0, very acidic because of the mineralogy. And the water temperature can range from 30 degrees to 108. So it is just the authors of this paper that I'm going to describe, when they were sampling these pools, you could see dead birds all over the place because of the fumes being given off in these different pools. So this is not a nice place. I mean, it's beautiful and there's water. You can see the water, but what does it mean if we have water? This paper was looking at polyextreme. So you have one extreme, let's say you have low pH, but you could have a couple extras, low pH and high salt. Oh, and one last thing, this area is kind of interesting, particularly because you wind up seeing structures in the surface that have analogs on Mars as well, in these plains in Mars. So you can have similar types of potential geology or geological formations that do have similarities on Mars. So people are really interested in seeing, okay, if we find something here, what might it mean on Mars? That's our analog. So when they sample from these pools at Danakil, you can see what happens when the water cools down. All the salt just goes, and it just completely falls out of solution and precipitates. I mean, these are amazingly high concentration salts with a lot of different metals in it as well, iron in particular. And I think manganese and a few other different metals are in high concentration. And what they find is that, you know, they sampled from what you consider like an extreme environment. Maybe it's just hot or just salty, but not too bad otherwise. And these polyextreme environments. And in the extreme environments, if these bars represent the abundance of different groups of microorganisms, particularly archaea, you can see in the extreme rather, there you get fairly decent abundances. The number of reads is what it's classified at, but just think about the higher the bar, the greater the abundance. In polyextreme is nothing. All the little signals you see here are contaminants, or they've hypothesized that, I mean, you have these regions in close proximity. There could be dust blowing in to the regions that also give the DNA. This is DNA based, by the way. So they go in, they just take the sample out and they extract all the DNA, if there is any. And that's the thing. The other thing, and this was also really telling, is you have to look at the structures that you find. So I want you to, we're going to play the last game of the day, okay? One of these pictures is only a salt. Which one is it? A, B, C, or D? Okay, we'll do a show of hands. Who thinks it's A? Okay, we got one for A. Who thinks it's B? Two for B. C? A three for B? Okay. And who thinks it's D? Congratulations, Pauline. Those are not cells. Those are minerals. Look an awful lot like cells. So when you see these microfossilized structures, got to be careful. You can tell this apart though, because they can use different techniques like spectrometry, Raman spectrometry, and you can see if there are organics in there, but it still does not mean that was a live cell. So to close off, one of the big questions that we have nowadays is, well, first let's look at Mars. We really are, and it's really actually kind of interesting to see how this perseverance is going to develop over time. So just some fun facts about Mars. A day on Mars, a sol, is 24 hours and 40 minutes. So pretty close. But a year is 667 sols, or 687 days, obviously because the orbit is larger. There are seasons on Mars because it's inclination. The tilt of the planet is not too far from the tilt that Earth is. I think we're at 14 degrees, or no, that's the difference between the magnetic pole and the North Pole. It's very similar. I'm losing my numbers now. But the thing is, the atmosphere on Mars is 6/1000th of the pressure that our atmosphere is. So it's 0.006 bars. So very, very thin atmosphere. The temperature can range from minus 133 up in the poles to 37 degrees. I mean, we can't get quite to 133 on this planet, but we can get pretty low. You can get minus 80, I think, in Antarctica. I think that's, I forget what the record is, but it's in one of the Russian stations down in Antarctica, where you got down to minus 70 or 80 or something. There is a hypothesis that there might be liquid water, but it's very unstable. It only appears very periodically that they can see so far. And most of the water too is going to be up in the polar caps. And then you might have, if there's any level of warming up or geological activity, which they don't think there is, it might be some ice under these polar caps, or at least remnants, or some, rather water under these polar caps. So the big question is there life on Mars? I've been waiting the entire lecture just to show David Bowie, I gotta say. But we've been looking for some time. So if you look at the missions that have come up, you can see over the years, these are the Viking missions from the 70s. That's my birthday, 76. Not to tell you how old I am, but I just did, so whatever. And then you have all these different missions that have occurred over time. The Russians were, I mean, that's the thing, just a little side note. It's interesting to see the race, the space race results nowadays between the Russians and the Americans, because a lot of this exploration wasn't necessarily being done because it was scientifically interesting. It was just to beat the Russians, at least from our perspective, and vice versa. So the fact that some interesting stuff came out of it, that feels good, but maybe not for the best reasons. So we can just walk through a little bit. So the Viking landers in '76, they were the first ones that really had these experiments on board to try to figure out if there's life. They had different ways of measuring organic compounds, and they actually had an experiment where they dumped in a nutrient solution. They took in a soil sample and added a nutrient solution to see if there's any organics or carbon dioxide being produced. And they did this also with C14, radioactive carbon. And they used a couple other methods as well to see if there is anything. And the result is questionable to negative, because when they measured the production of these organic compounds, they found that there was organic compounds there, and they found that carbon dioxide and everything was being produced. But the thing is, in Mars soil, there is a ton of perchlorate. Perchlorate is particularly nasty. The soil of Mars is incredibly oxidized because of all the radiation. So that's actually a big hazard for astronauts, because that soil is toxic. So if you come back with a lot of Mars dust on you, that is extremely unhealthy. So that is a concern. It also means that it's not looking good for life being available, because that stuff is also going to be nasty for the microbes. But we go back to Earth again. There are regions on this planet in the Atacama Desert in Chile that also have high perchlorate concentrations in the soil. So there's a lot of active work looking at the microbes that might be living in this region as well, to see what that might be like on Mars, if there is anything. Another possible bit of evidence are these asteroids. So there's actually been a transfer of rock between Earth and Mars throughout history, geological history, because you get asteroids striking Mars or striking Earth, stuff goes up into space, and over time, you can get a transfer. And if they're large enough, the asteroids can land intact enough where you can tell by the geology that that did not come from Earth, that came from Mars. At least that's the hypothesis. It's the most likely candidate or reason. And there's one particular asteroid, asteroid, one particular meteorite is the more correct term, where they found it in Antarctica. And after looking at it carefully, they found these structures again, and some of them had different types of organic material surrounding them. So there were organics on it. And then they looked further and they find these different sort of fossilized little potential cells. But we also don't know if that is contamination, because this meteorite's been lying in Antarctica for hundreds and probably a million years, hundreds of thousands of years. I think they estimate it as like a couple 100,000 years old. The other thing is, if you have this exchange between Earth and Mars, let's say we find rocks on Mars that have microbes in them. Did they come from Mars? We won't really know. So it could be that Mars seeded us with life, or we seeded Mars with life, and then it died out as the atmosphere got blown away by the sun. Now, the last little bit of evidence that's interesting is the methane release. That this is Opportunity rover, 2006, I believe. And as it was exploring and taking measurements, it found that there are these methane fluctuations in the atmosphere that are seasonal, they're periodic, and they occur at a very large, planetary scale. This is not unlike what you see with a lot of planetary processes on Earth in atmospheric gases, or, for example, cyanobacteria, things like that, global processes. So maybe there are microbes that are producing the methane, but then it dies back in the late spring, but then increases in late summer, autumn, and then goes back in the winter. So is there a seasonal blooming of life, basically, that's producing methane? So there'd be methanogens, which again are archaea. So it's a neat hypothesis. But of course, there are many different processes that could also lead to methane production that are abiotic. So you can have, I won't rattle through all of them, but you can have a variety of things because there could be organics that are being hit by UV and it just gives off methane abiotically. There could be all different kinds of photochemistry. There could still be maybe microbes in the water underneath the subsurface that we're unaware of that are producing, and it's outgassing into the planet periodically. And these other geochemical processes as well, water with olivine, again, producing methane, but it's still not life. So finally, is there life on Mars? This is something that Perseverance is going to look for. It's got a rather advanced array of instruments, really high-tech spectrophotometers and spectrometers, and it's targeting this crater where you can see, at least geologically, it was highly likely that there was water flow here a long, long, long time ago, with a river delta, an ancient lake bottom. Possibly this area could be something like that Donakill Depression, this Niels Planum. They're interested in that. So it'll be interesting to see what they find. And yet that is not the last place we should be looking either, because there's a lot of scientists that are arguing, screw Mars, let's go to Saturn, because there's much less of a chance that there's been an exchange of material between Mars and Earth and Saturn. And you have this moon, Enceladus, which is, you know, it's not that big. It's only the size of the UK, but it makes this huge vapor ring, this E ring it's called, around Saturn. Because of the geysers, it's blowing off. This is water and salt that's coming off of this moon. And it makes these rings. This is Enceladus right here within this hazy ring. And there's a lot of talk about sending a mission where they could actually put like some probe that melts into the ice to see what they can detect. But we got a ways to go before that.